Welcome to number 7 in our series from Drug Free Australia on the cannabis population studies. Again, we're with Dr. Stuart Reece, the world authority on those population studies. And over to you, Stuart. Thanks.
Thanks, Gary. Thanks for having me. Yeah, I'd just like to present a summary, sort of shortish summary talk of some of the main points we've talked about. And it's a bit of a long title, but it says downstream sequelae. That just means the consequences of genomic and epigenomic. That's the hardware and software of the genes. Cannabinoid toxicity, including food chain contamination. So this is a summary of our series of discussions. And this is our book, Epidemiology of Cannabis, which will be out shortly, which discusses it all in detail.
Which was due in March originally, but has been delayed a little.
That's right. There are no particular conflicts of interest to declare. So we're talking about cannabinoid neurotoxicity and genotoxicity. We've talked about mental illness, autism, cancer, congenital anomalies, and aging. We've particularly featured the brain, limbs, chromosome, birth defects, and cancer. 8 alphabets of life are disrupted by cannabis. The genomic code, 59% of the chromosomes are directly implicated. The epigenomic code, at least 20,000 hits. The histone code we've mentioned. Some of these we haven't had time to go into in detail. The phosphoproteome, the activated proteome, the proteins that get phosphorylated and activated in the cell. The acetylproteome, which is another signaling system. The tubulin code, we also haven't had time to get into, but we show that cannabis disrupts tubulin in many important respects, which clearly disrupts the tubulin code. The sugar code we haven't discussed. That's another discussion. We've published on that. And mitochondria we've mentioned at various points, being key to the maintenance and health and well-being of the genes in the epigenome. So by way of summary, cannabinoid neurotoxicity, cannabis is closely related in a space-time association and fulfills the criteria of causality for both all 4 mental illnesses, which we discussed, and autism. So that's in adults, young adults and adults, and in children after prenatal exposure. Numerous mechanisms are involved, particularly disruption of the morphogen gradients that are responsible for weaving of the brain together. Autism-related genes as they're triggered epigenetically, and the epigenome. So there's 3 key things, and the synaptopathy. We also talked about the disruption of the synapses or the connections between the cells. This is a photograph through a microscope of normal human sperm, and this is sperm on cannabis. You can see that's a pus cell in the middle there, and some of them have got too many tails and bits of head halfway down the neck piece, and bent, shriveled up heads, and damaged tails, and multiple tails, and multiple heads, 3 heads, 4 heads there, 3 heads there. This is a grossly disrupted sperm structure and function. This is the same thing looking at sperm head histone assays. This is a normal histone stain here. You can see after cannabis, it's completely different. This is a normal stain with this technique, and it's completely disrupted here, and even more so in this one. We showed that a cell division after cannabis is disrupted, whereas cells normally divide into 2. It's called bipolar cell division. You can divide into 3, 4, 5, and these chromosomes get left behind. Bits of chromosomes don't separate and become broken, traumatically, physically broken when the cell divides. This is mouse sperm exposed to cannabidiol, and you can see that the head is down here, the tails are damaged, the head's grossly deformed, all sorts of problems, major problems with these sperms exposed to cannabidiol. Mouse sperm, we looked at chain 4 chromosomes stuck together, ring chromosomes, chromosomal fragments, a ring there and a fragment there in this study. We looked at the study by Stencheva and saw all the DNA breaks that are clearly illustrated there. This is a study by Zimmerman, And this is what we talked about, the cannabinoid structure. As you can see, we'll talk about the structure perhaps first. This slide's a little bit busy, but there's THC, and that's the levotyl structure. And you can see the levotyl structure is here. This is the C ring of the THC. There's cannabichromene, and there's the levotyl structure, cannabigerol, the levotyl structure, cannabidiol, the levotyl structure, and cannabinol with the levotyl structure. So it's the C ring, which is a common structural component of all these different cannabinoids. And so then you come to this chromosomal translocation assay. They've looked at the number of chromosomal translocations. They've got controls here. It's about 1% saline, and that's an organic solvent commonly used in the lab, DMSO. This is a positive control, mitomycin C. This is a cytotoxic chemotherapy agent. It's about 7% translocations after mitomycin C. And THC, cannabinol, and cannabidiol have 4, 5, and 6% translocations in the region of the positive control. So this is chromosomal translocations. This is severe genetic damage, and the chromosomes are up to 300 or 400 megabases long. So this is at the megabase scale, testing positively with the mitomycin C in this assay. So there's 2 points there. Firstly, that these different cannabinoids, THC, of course, psychoactive, cannabinol and cannabidiol not psychoactive, but testing is strongly positive, almost as strongly positive in this assay as the known carcinogen mitomycin C. And the levotyl, you can see in all these different cannabinoids exactly how it forms part of the structure of these different cannabinoids. THC psychoactive, these ones not psychoactive. But this is the genotoxic moiety, and you can see it's related. It's an aliphatic derivative of phenol, which is hydroxylated benzene ring. So all the cannabinoids are involved. That's a very important point. Levotyl and THC and all the others. Delta 8 THC, Delta 9 THC, Delta 10 THC, cannabidiol, cannabigerol, cannabichromene, cannabinol. I might just point out here that the Delta 9 and Delta 8 refers to the plane of some of these hydrogen bonds compared to the plane of the molecule. And the Delta 8, Delta 9 just talks about which of this is 8, 9, 10, round like this, whether the molecule is up or down. So the Delta 8, there's a lot of confusion in America about Delta 8, Delta 9, Delta 1, Delta 10, but they're very minor modifications of minor reactive groups around the THC molecule. So that they don't affect the genotoxicity, which is related to the central levotyl structure. We talked about the importance of morphogen gradients, bone morphogenetic protein, the Wnt proteins, and sonic hedgehog to specify exactly where in the tissue these cells are going to develop. This is the spinal cord, but the same thing applies to the brain and the heart and the limbs and all the organs as they develop. And the gradients of these, the disparate gradients, the conflicting gradients, dictate exactly where these cells are going to form and how they're going to behave. Now, direct chromosomal damage. If you look at ALL, the chromosomal trisomies, and testicular cancer, the trisomies we mentioned, 18, 13, and 21, the chromosomes that are known to be damaged in testicular cancer, the chromosomes that are known to be damaged in acute lymphoid leukemia, these are the chromosomes that are relevant to these three diseases. And if you add the lengths of all of these these are the lengths of the chromosomes, up to 246 megabases for chromosome 1. You get 1,765 megabases. Now, the total length of the human genome is 3,000 megabases. It's 3 billion bases long, which is 59%. So 59% of the chromosomal length of the human genome is known to be directly involved in cannabinoid genotoxicity.
Just with those chromosomal trisomies, some more popular names that we might understand them by.
Down syndrome, Edwards syndrome, Patau syndrome.
Okay, thank you.
Yeah. So So is that clear, Gary? When you add together all the chromosomes that have been known to be implicated in those diseases, you get nearly 1,800 megabases of the 3,000 in the human genome, which is 59%.
And they're all susceptible. Yeah.
So the hallmarks of cannabinoid aging is syndromic pattern of illnesses, cardiovascular age acceleration, endocrine disruption, mitochondrial inhibition, altered DNA methylation, which is heritable, neuroinflammatory mental illnesses, because the brain itself gets inflamed. These are all key hallmarks of aging that we're talking about.
Right.
Cirrhosis is linked with cirrhosis and inflammation of the liver, damage to eggs and sperm, genotoxicity, cancer and inheritable cancer, and genotoxicity, congenital anomalies. These are all aging-related stigmata or hallmarks. It inhibits telomerase, which we talked about in the aging discussion. It damages chromosomes, causes immune stimulation, causes chromosomal damage, impaired respiratory exchange ratio in the mitochondria, reduced ADP production, extra chromosomal DNA circles, that's an age-related change, double-stranded DNA breaks, reduces lamin B in the nuclear membrane. This is what is seen in progeria. You know those people who are old when they're 12 years old? This is the change seen in those. And it's also linked with mortality. So there's these 20 hallmarks of aging, which are discussed further in the aging discussion, which show that cannabinoids drive aging. In particular, the sperm and the oocyte, they've been mentioned at multiple points in these discussions. Together, of course, form this fertilized zygote, which means that the zygote, if the sperm is aged and the oocyte is aged, the egg and the sperm are both aged, then the zygote must be aged by definition from the moment of fertilization. But the zygote is actually aged from before fertilization, because the sperm and the oocyte are aged before they get fertilized. So the zygote is actually aged before it even is a zygote in the pre-fertilization stage, if that even makes sense. And we talked about mortality, the greatly elevated mortality. It's almost 30% over 30 years. And many studies document the increase, a death rate from cannabis, which can happen through many pathways: schizophrenia, suicide, heart attack, cancer, congenital anomalies. We said that cannabis inhibits the 8 alphabets of life, which we talked about earlier. We said that cannabis affects mental illness, autism, cancer, congenital anomalies, aging, epigenome, transgenerational, exponential effects, and it's all the cannabinoids are involved because it's the 11-tol nucleus is the deal. And we talked about the interaction between the genome and the epigenome, so that the hardware damages the software and the damaged software damages the hardware. All these things are detailed further in our book. And so this has been a discussion of the downstream sequelae of genomic and epigenomic cannabinoid toxicity, including food chain contamination. Thank you so much for your attention.
Thank you, Stuart.